Recently, Lau et al. Have studied the use of yeast surface display in engineering a soluble and more crystallizable construct of HPK1, which is a protein involved in the modulation of T-cell mediated tumor responses. So it is a fairly difficult protein to study as it does not like to be crystallized, as it is not very soluble and also has very poor expression levels in recombinant systems. This is why Lau et al. Decided to use a yeast surface display technique to try to find more soluble and crystallizable variants, specifically of the kinase domain, which is residues 1 through 346. Their workflow was to start with an analysis of the spatial aggregation propensity. They did this in both BioVia Discovery Studio as well as in Bioluminate. And these analyses resulted in 8 predicted positions on the surface that were prone to aggregation. You can see that figure on the left shows these positions. They went ahead and mutated all of these residues in all possible permutations to various polar residues, and this gave rise to over 1.6 million possible mutants. So they went ahead and they generated a library of all of these mutants and amplified all of that DNA. And then they performed 4 rounds of fluorescence activated cell sorting, known as FACS, to select for the most highly expressed variants. And you can see the results of this in the figure on the right of the slide. The top row here is just cells expressed in rich media, and the bottom is in minimal media. So after this 4th round of sorting, the enriched variant plasmids were recovered and purified and sequenced. And the sequencing showed that there were 16 variants that had unique combinations of mutations at 2 different aggregation-prone sites. So large-scale expression was carried out for these 16 variants. And then they were purified and examined by SDS Page. Only 11 of these variants were found to be soluble, so those were proceeded with. And to determine which mutations in these 11 variants were responsible for the heightened solubility, one by one, the mutations were reverted back to the wild type. And then SDS Page analysis was performed with those resulting mutants and showed that 2 positions were essential in the heightened solubility, those being 221 and 225. So these findings led to the design of the mutant L221D F225E, which was actually successfully crystallized at a very high resolution. So this study shows that yeast surface display is a very handy tool in trying to construct more crystallizable variants of these relevant proteins.